What do you imagine when you think of the word mutant? Do you think of X-Men and some really awesome ability? Well, in reality, mutations are not that glamorous. The genetic information in the cell is stored in the form of DNA. The nucleotides from the DNA are transcribed to their complementary form of RNA, which is then read as codons, or groups of 3 to code for specific amino acids in the larger protein. These proteins make up or influence different traits. In DNA, the base adenine A goes with the base thymine T, and the base cytosine C goes with the base guanine G. When the wrong base matches up, mutation in the DNA occurs. So, a mutation is the change in the cell's DNA that leads to abnormal protein production. It could be due to external factors or internal factors, like a mistake during DNA replication in interphase. Interphase is the stage in which the cell copies its DNA in preparation to divide during mitosis. While most mutations are neutral, that is, they neither have a positive or a negative effect, some mutations can be harmful. Very few mutations are actually helpful. So what are the different types of mutations? Substitution. This means you have the wrong base matched, or there is a change in a single base. So instead of an A with T, one can have an A with G. Sometimes substitutions may not affect the protein structure. Such mutations are called silent mutations, and sometimes they may change an amino acid coding codon to a single stop codon and cause an incomplete protein. This can seriously affect the protein structure, which may completely change the organism. Example of a substitution mutation is sickle cell anemia. Insertion. Insertions are mutations in which one or more bases are inserted. The number of base pairs inserted can range from 1 to thousands. An example of insertion mutation is Huntington's disease. Deletion. Deletion are mutations in which one or more bases are lost or deleted. The number of base pairs deleted can again range from 1 to thousands. 22q11.2 deletion syndrome is an example of deletion mutation. Frameshifting mutation. A frameshifting mutation occurs when a protein is drastically altered because of an insertion or a deletion. Unlike deletion and insertion, substitution mutation occurs at a single point within the DNA strand and usually only affects the codon that contains the altered nucleotide. Hence, substitution mutation is less influential than the insertion or deletion mutation in most cases. Why is it that some of us can enjoy a glass of milk, while for others, it's not such a winning proposition? Let us look at how cultural changes have resulted in a mutation that allows some people to digest milk. From the time our species evolved some 200,000 years ago, we have lived as hunter-gatherers. About 10,000 years ago, people started domesticating animals for food and cultivating crops. These cultural changes had some biological impact on our species. One such thing is our ability to digest milk as adults. Milk. All of us can digest it as babies. In fact, producing milk for babies is a key trait that distinguishes mammals from all other animals. The main sugar in milk, lactose, can't easily pass the intestinal wall, so cells here make an enzyme called lactase. Lactase breaks lactose into glucose and galactose. These simpler sugars can then enter the blood and be used for energy. After the weaning stage for mammals, there is a downregulation in the production of lactase until they essentially lose the ability to digest milk and become lactose intolerant. When an adult mammal drinks milk, the lactose goes undigested straight through the small intestine and to the large intestine. Here, bacteria consume the sugar and cause gas, diarrhea, and cramps. The only mammal species in which some adults can drink milk is humans. 30% of adult humans can digest lactose. This minority is called lactase persistence, as their ability to produce the enzyme that breaks down lactose persists well beyond infancy and childhood. How did this happen? Let us look at the genetic foundations of lactase persistence and the mutations that keeps the expressions of lactase gene permanently on. The LCT gene is located in the long arm of chromosome 2. It is responsible for encoding the enzyme lactase, which is produced in the villus enterocytes of the small intestine. A point mutation in the DNA occurs that allows for lactase enzyme to be produced in adulthood. The mutation occurs at the nearby MCM6 gene. The MCM6 gene is a lactase regulating gene. In people who can digest lactose into adulthood, the MCM6 gene has a non-coding stretch of DNA, called the regulatory element, that has different single nucleotide variations that allow continued expression of the LCT gene past childhood. The two most well-known variations are when cytosine is replaced by thymine, or when guanine is replaced by adenine. Without these variations, expression of LCT gene declines. The prevalence of lactase persistence is highest in human populations that have a history of raising livestock for dairy consumption. These include Northern Europe, Southern Sudan, and Northern India. Lactase persistence is one of the most recent examples of social and environmental effect on genetic evolution.